Alright, so we're going to do our trial run at using uh, online lecture for Biology 1510. We're going to lecture on chapter 22 in the Raven textbook, which is called The Origin of Species, kind of our last chapter in our series on evolution for this course at least, although obviously we talk about it throughout the entire course. So this is what we would call the macroevolution part of the topic of evolution. Macroevolution refers to changes that occur above the species level or at taxonomic categories above the species level. So how do we get to have distinct species or families or even classes and orders of different species? So what we have to look at here is, first of all, two things. The fact that species are comprised of different populations, different species that live in the same place, maintain their identity as separate species. They don't blend together into multiple species. You can have several hundred or even thousand species living at the same place at the same time, and for the most part, they will never ever breed together successfully. Very rarely will that actually happen, as far as we know. So on one hand, we have this distinctiveness of species living in the same locale at the same time. On the other hand, populations within the same species very often live in different places can actually have some degree of connectivity and interact back and forth between two subpopulations of a species. So these are what we call sympatric species. Sympatric species, by definition, occur together. They are, like I said on the previous slide, distinctive. They do not typically blend together or hybridize very often or very successfully with other species. They tend to be phenotypically different, at least to us. Now, this is a very, very critical point because just because two things may look similar to us doesn't necessarily mean they look similar to each other. Our focus is on the visual. Okay, so other species may focus on auditory cues, chemical cues, behavioral cues, things aside from physical appearance. Perfect example of this is the two organisms on this slide. We have on the one hand up here on the top, we have a chimpanzee. On the bottom hand, we have something known as a bonobo, which up until the past, decade or so, maybe a little bit longer than that, were thought to be the same species. But upon further observation, what people noticed was even though they look very, very similar, they actually are behaviorally quite different, and these two, what we now refer to as species, will not interact with each other. They actually are very different behaviorally. Chimpanzees, on the one hand, that's our upper right picture here on the slide, the chimpanzees are a very, what you might refer to as an aggressive or warlike species. When they have difficulties between adjacent populations of chimps, they go to war, they kill each other, they can be extremely violent. Yes, they look to be very human-like, and behaviorally they are more human-like. They prefer to settle their problems with violence. On the other hand, down here on the bottom right picture, I actually chose this picture because I feel it kind of illustrates kind of the overall personality of what a bonobo kind of behaves like on a day-to-day basis. They are very much a passive type species. When they have problems, when they have difficulties, when they have stress, they don't go to war. Instead, they choose to go into what you might say is the opposite. They go into copulation. They solve their problems with sex. So they'll have group sexual encounters. They will have male to male, male to female, female to male encounters, and it just seems to be part of the day-to-day activities. They almost seem to enjoy having this interaction among members of the same group or the same tribe, or even between tribes. It's a way to resolve conflict in some ways. On the other hand, other species may use different parts of the habitat. You may have two species which are completely reproductively compatible, but because they live in different habitats, maybe one lives in the forest, one lives in the field, one is active during the daytime, one is active during the nighttime, they may not actually encounter one another, and they may not, in fact, breed together with one another if they simply don't run into each other. We talked about behavioral differences as well. So the thing to remember here is that phenotypic differences, physical differences, don't necessarily always define species. They can be behavioral, they can be spatial, they can be ecological, they can be temporal, they can be lots and lots of things which prevent different species from breeding together. We can even go to what we call a subspecies level, where in this case we have actually several different subspecies of milk snakes, which occur more or less east of the Mississippi River. This line that runs right down through here is the Mississippi. There's a little bit west of that, but for the most part, they're east of the Mississippi, from the dark green all the way up here to these two yellow patches, to this blue patch, to the purple patch down here in the southeastern U.S. They are all completely reproductively compatible. So when you find transitions from one subspecies to the next, from the red milk to the scarlet king snake up here, from the sort of middle ground here and the middle ground up here, you actually find hybrid varieties of these different subspecies. So they are reproductively compatible, but they are separated geographically. They're almost like having separate species, but they're not quite there yet. It's like they haven't been apart long enough to have developed true perfect species isolating mechanisms. They still are compatible, and even though they may have started to look very different, they start to take on a different phenotypic appearance, maybe to match their habitat, but when you bring them together, they can and actually will produce viable offspring. All right, so the most traditional definition of what a species actually is, all the way back on day 1 of 1510, all the way back in August, was that species are defined as groups of organisms who are reproductively isolated from other such groups, or even more specifically, groups of actually or potentially interbreeding organisms which are reproductively isolated from other such groups. The key to that definition is reproductive isolation. Species mate only with each other, they do not mate across species boundaries. That concept works great for a lot of species. And in fact, when you have reproductive isolation where two different populations live in different places, different habitats, different challenges, there is a very good chance they may develop reproductive isolation if you give them enough time and if the environments are different. But this definition doesn't work in all cases, and we'll get to that here in a few minutes. Reproductive isolation, the development of reproductive isolation is not necessarily the only way to define a species. So for example, species that don't reproduce sexually. How do you know if they breed across species boundaries if they don't breed across the organism boundary? That definition falls apart. It's meaningless when you're talking about things like bacteria because they don't reproduce sexually ever. So how are you going to know if they can produce hybrids when they don't have that capacity for sexual reproduction in the first place? So when we talk about reproductive isolating mechanisms, we could have things like geographic isolation, simply separated by space. Maybe there's a mountain range or a river or even an ocean in between two populations, which divides them efficiently enough that they actually eventually develop reproductive isolation. They never encounter each other. You could have ecological isolation where two species might be reproductively compatible, but if they feed on different plants or feed on different parts of the same plant or feed in different habitats, again, they may never actually run into one another and be able to produce offspring. Behavioral isolation. Maybe you have species such as this one, which apparently is devoting its time and energy into some sort of costumed reproductive dance, would only attract members of that same species. Whereas other species would have different behaviors, and they would not be attracted to the species, nor would the species be attracted to them. So they actually have to go through some sort of a call or mating ritual or dance or some sort of very standardized behavior to ensure that they are members of the correct species before mating. Temporal isolation, again, is like isolation in time. If you have a species that flowers, male flowers become reproductive in May, and the other species, female flowers, become reproductive in August, chances are they're probably not going to be able to cross-fertilize, even if they are truly reproductively compatible. If they don't breed together naturally on their own, they're still considered to be separate species. The last one is mechanical isolation. So all the ones we've talked about so far are what we call pre-zygotic isolating mechanisms. They are ways of keeping species apart as distinct species before a zygote is actually formed. These are basically pre-mating, pre-formation of a zygote type isolating mechanisms. Mechanical isolation is another one. Mechanical isolation is a situation where, as I like to say, the parts don't fit. So male and female genitalia don't actually go together. If that's the case, then you cannot have successful fertilization. And then your sort of last line of defense against hybridization between different species is the fact that the gametes may not be compatible. Perhaps sperm cells don't recognize egg cells of the other species. Perhaps egg cells of one species don't accept sperm cells of a different species. So if the gametes do not fuse, if fertilization does not happen, you still in fact have reproductive isolation and you in fact have different species. It could be that maybe you have species in the same place, living in the same habitat, behaving compatibly enough that at least they'll get together, they're active at the same time of day, the parts fit successfully. If mating occurs and fertilization fails, you still consider them to be separate species. On the other hand, you could also have post-zygotic reproductive isolating mechanisms where after a zygote is formed, the hybrid is either inviable, meaning it's somehow weak or sickly or just generally not able to survive, or it could be that it's infertile, it could be sterile. So if you were to jump back to that last slide, the one that had the guy with the fun-looking sunglasses being pulled by a team of two mules, mules are a perfect example of a post-zygotic reproductive isolating mechanism, as are ligers. You've heard of ligers before, right? So they're half lion, half tiger. They are perfectly compatible at a genetic level. You can actually breed them together artificially and produce a liger or a tigon, depending on which parent is the male, which parent is the female, which species is the male, which species is the female. They will breed together. Now, the mules, on one hand, are produced by a combination of horse and donkey. And when those mules are produced, they are sterile. They are big, they are strong, they are certainly viable. They're used or were used to do a lot of really heavy work on farms, but at a reproductive level, they are completely sterile, and so horses and donkeys are considered to be separate species. Lions and tigers, on the other hand, are considered separate species because in the wild they do not encounter each other naturally. Lions being from Africa, tigers being from Asia, lions being savanna open habitat species, tigers being jungle species, lions being gregarious or they live in social groups, tigers being solitary. They just simply don't get together to produce offspring, even though their gametes are compatible. This is an example of behavioral isolation. This is one of my favorite slides in the entire class because it gives me a chance to talk about boobies. Yes, these are boobies. These are blue-footed boobies as opposed to the also common brown-footed boobies in certain parts of the world. And so what we have here is a male on the left working very, very hard to try to convince this female over here to mate with him. So he's going through a very ritualized and very standardized series of moves in order to attract the affections of the female, at which point she might choose to mate with him. The female, on the other hand, being the choosier of the two sexes in this species, gets to pick among all the available males and pick the one that she thinks might have the best genes. Of course, this may not be a conscious decision, but based simply on the male's reflection of his own fitness or well-being or goodness, his worthiness of being a mate, and sort of fostering the offspring of those females. Big change from boobies to lacewings here. You may also have other types of signals like visual ones in the birds, sound productions in things like lacewings. These are green lacewings in this picture here. These are a very, very common species. They are insects. They're in the order Neuroptera. Very common in this part of the world. In summertime at night, you'll have these flying around your porch lights. They actually go by a series of clicks. And so different species will be attracted to different frequency clicks and different amplitude clicks of members of their own species, but not anyone else. Chemical signals may involve things like pheromones, smell. So a pheromone in general is a way of one species or one individual communicating with another member of the same species. So you release a chemical that might be involved for, in this case, mating. Other pheromones could be used for attack or for trail following or for food gathering type purposes. Mating pheromones are relatively common in the animal world. They're chemical signals sent by one member of a species, one sex in the species, to attract members of the opposite sex. And some species may be even using electrical signals to perceive the presence of a potential mate as well. As far as temporal isolation, this is another example from your book. It's a species called wild lettuce. It's essentially a weedy and somewhat invasive exotic species in the southeastern U.S. Two species here are perfectly compatible reproductively. If you bring them together in a lab setting or in a greenhouse, they can breed together, they can produce offspring that are an intermediate between the two species. However, flowering time for these two species is quite different. One flowers in spring, the other flowers in summer, so even though the gametes are compatible, the gametes typically aren't together at the same point in time throughout the year, so they are still considered to be separate species. Mechanical isolation shows up very, very commonly in insects. So insects very often will have very, very elaborate genitalia that will only fit members of the opposite sex of their own species. Bees, for example, carry pollen of one species of plant in certain places on their body. And so it's very, very important for the plant to position its anthers or male flower parts to be able to deposit pollen on that particular part of the honeybee. It's also very important for the female flowers of that same species to be able to receive pollen from those honeybees males in order to be successfully pollinated. They're extremely specific. One species of bee will only go to one species of plant at a certain time of the year. There's a very low likelihood, even if two plants are compatible, of that bee visiting the same flower at the same time of year, carrying pollen in the same way so that it's deposited on the same part of the female flower of the opposite plant. So the odds are pretty long in that case of successful cross-fertilization happening. What happens if you have external fertilization? So these two species down here are frogs. They look like they could be wood frogs. It's kind of hard to tell from this picture, but they might be wood frogs here, Miranda sylvatica. So we've got these two wood frogs. This is a male on top here. This is a female, right? And so the male is engaged in what's called amplexus. So that would be A-M-P-L-E-X-U-S, amplexus, where the male hangs on sort of around the waist or the chest of the female and waits for her to deposit her eggs, as this female has done here. Now this happens in the water. When the female deposits her eggs out of her body, the male is going to deposit sperm over those eggs and form gametes, which eventually turn into eggs, and when fertilization happens, the zygote and embryos from that point on. Now you can imagine in a pond in the springtime, there are all different kinds of species of all different kinds of frogs doing the same thing kind of at the same time. It wouldn't be unusual to go to a pond in the springtime, in the peak of breeding season, and find 4 or 5 species of frogs all breeding externally in the same place. One big sort of a frog orgy in these springtime ponds. It's actually kind of cool to hear this when you see all these frogs calling back and forth and all the different types of calls even each species has. But the problem that comes up is if you have 5 species of males trying to fertilize 5 females of those different species, what prevents the sperm cells from one male from cross-fertilizing eggs of a different female from a different species? Well, just like we said before, those gametes may not be compatible. His sperm may not be attracted to eggs of a different species, and eggs of a different species may not accept sperm of a different species. So even though the gametes are in the same place at the same time, fertilization, gamete formation, egg, embryo formation may not occur. Plants are the same way. Plants are very good, or at least better, at hybridizing than our animals, but even they have some degree of discriminance when they're accepting pollen from a different type of plant. So pollen tubes, the way that the actual sperm cell gets from the stigma of the plant and the style of the plant down to where the ovary and the ovules are, the eggs, is through a pollen tube. Sperm is deposited through a pollen tube to where the ovules are, where fertilization takes place. But female flowers on female parts of plants will only accept pollen in pollen tubes of the correct species. Even though pollen, which is carried on the wind and may land anywhere, you could have pollen from pine trees landing on flowers and oak trees, it's not going to fertilize those oak trees. They're not compatible. So zygote formation and growth isn't going to occur. So post-zygotic isolating mechanisms, again, here's another group of frogs. These are leopard frogs. And again, a huge, huge chunk of North America, all the way up into Canada, which is sort of unusual for ectothermic organisms. These frogs occur over a huge swath of North America. And these species, even though they occur adjacently, even though you have, right here in southwestern Michigan, you have all 3 species kind of occurring almost together at the same time, they don't hybridize. And even if you bring these species together in the lab setting, they will not hybridize. The gametes are incompatible. And even when hybrids do form, on those rare cases when you do get hybridization between 2 species, the hybrids don't work. They could be sterile, they may not have the correct sex organs, they may not be able to produce gametes, so even though they're breeding, they might be making a zygote, they're not going to be able to breed, and so therefore those 2, in this case actually they are subspecies, are not going to be able to produce offspring. They're going to maintain their identities as separate species. Now, I mentioned before that the biological species concept does have its flaws. There are some problems with this concept, right? So, for example, in one case, or one study that was done, plants in particular are not very well defined by genetic isolation. There's a lot of different plants that will hybridize with a lot of other species of plants. If you look at oak trees in this part of the country, and south of here, oak trees will breed with all different kinds of other species of oaks. There are 200 or 300 different hybrid varieties of oak trees that are out there, and they're really difficult to identify because they're actually crosses between 2 oak species. Birds are very good at hybridizing in some cases as well, and what we perceive as being different species may not in fact be different species. They may actually be one species that happen to look sort of different. So a lot of hybridization even in the animal world when it comes to birds. Asexual species. Hard to tell if they can breed with other species if they don't even breed with members of the same species. So bacteria, other asexual species, very difficult to define them as species using this species concept, the biological species concept. Species that hybridize in captivity. I mentioned lions and tigers before. They breed in captivity, but they don't breed in the wild, so what do you do with those? They are technically reproductively compatible, but they don't do so in nature, so are they the same species? Are they different species? It's kind of an arbitrary line to draw. What about fossil species? What about species that have been extinct for 300 or 400 million years? It's really hard to see if they breed together if they're isolated only in rock. They're not going to breed with anybody, right? So for those species, the biological species concept is completely meaningless because breeding does not occur, period. So what one species can breed with or not almost becomes arbitrary. Another way of looking at species is what's called the ecological species concept. And so in this case, we're actually going to maintain species identity based on what kinds of activities the different species do in the environment, based on the niche. So niche is a very, very important term in ecology. A niche refers to a species' ecological role in the environment, or their ecological job in the environment. Right? So in this case, you've got species that are going through stabilizing selection, like we've talked about in class, where you're sort of selecting for the middle of the bell curve. The average type is favored. And at the extreme ends of the curve, the weirdos, the ones that are very extreme, those are selected against. And so if you have two overlapping species, niches, say in seed size or host size or something like this, if the two species conditions or resources that they require overlap, you may get hybrids, but those hybrids may not be good enough or specialized enough to do very, very well on one food item or another one. So in that case, the hybrids or the overlapping parts of the curve are going to be eliminated, thereby maintaining species integrity between those two different groups. Cladogenesis or cladogenesis is a situation where you actually have an ancestral species dividing up into two descendant species, right? So this species formation or cladogenesis, the heart of macroevolution, is how exactly do we get to have reproductive isolation, whether it's prezygotic, postzygotic, or otherwise. So what we're going to be looking at next are the concepts of allopatric speciation and sympatric speciation. And then we're going to take a break. I'm going to cut off for a little while. I'm going to start a second file so this one doesn't become too unwieldy. I've been going for just about a long enough amount of time on this lecture to be able to stop and move on to the next chunk of this lecture. All right, so species that are isolated, species that are occurring in different habitats whereby they don't actually ever run into each other, can become reproductively isolated. If they're living in two different habitats, they may start to change to fit their habitats. Natural selection occurs, evolution begins, and eventually, just by chance, you may actually develop not only physical isolation, but also reproductive isolation. And if those two species stay apart for long enough, even if you bring them back together, they may no longer breed anymore, at which point, of course, they have become different species because they are reproductively isolated. Right? So the easiest way to imagine how speciation could occur, how you actually get to have reproductive barriers between species, is when you have a physical, geographic type situation. If that geographical isolation is not complete, if there are individuals kind of leaking across the boundary, going back and forth between the two populations, reproductive isolation may not occur. So the more effective the isolating mechanism, the quicker the evolution in the two different species is going to happen. So connectivity is going to retract or detract from the isolating process. The more isolated two populations are, the more quickly and the more easily they might turn into two separate species. All right, so here's a case of what we call character displacement. Character displacement is where you have species which, when they live together, look very, very different. But when these two species occur separately from one another, they tend to look very, very similar. All right, so picture two birds living with a range that's side by side with other birds. We've got the pied flycatchers here and those that live down here. And in these two sort of ranges, the dark green and the yellow, these two subpopulations look very, very different. And the reason they look very, very different is they are beginning to evolve to fit into the different environments where they live. Different conditions, different habitats, different plants, different camouflage, different predators, they begin to look different. Where you've got species that do not co-occur, do not share a boundary, like the pied flycatchers in the north and these two in the south, they're going to tend to look relatively similar because now they're no longer competing with each other for resources. They can sort of go back and maintain that similar appearance between those two species or subspecies. Another situation that occurs when you have isolation between two populations is that of genetic drift. So genetic drift, as we've seen before, is a potential way to have speciation, or not necessarily speciation, but evolution happen, where you have random chance changes in gene frequencies, especially in small populations. So in small populations, there's a much greater likelihood of random things affecting those individuals which you have. Very, very good example of this, and at this point I haven't lectured on this, but I imagine I probably will lecture on this. I'm sort of predicting the future from the future, is that cheetahs, for example, went through a massive, massive die-off, 30 or 40,000 years ago. They actually went through a feline version of our version of AIDS. They went through what's called FIDS, or feline immunodeficiency syndrome, and 95 or 90% of the cheetahs that were on Earth went extinct. The only ones that survived were the ones that had some degree of resistance to that FIDS virus. The species that repopulated or recolonized the species are all descended from a very, very small number of these surviving cheetahs several thousand years ago. So therefore, they're all very genetically similar. There really aren't very many alleles in cheetahs as a species because most of different versions of those genes were lost 40,000 years ago when most of their species died off. Another good example of this is founder effect. So classic example of founder effect is where you have maybe half a dozen individuals of a species that get blown out to sea during a storm. They somehow manage to land on an island, they colonize the island, and they start to breed. Now those 5 or 6 individuals which represent the ancestral population on the mainland, because there are so few of them, will not very likely have all the alleles that were present in that mainland population. It's going to be a small sampling or a subset of the alleles that were in the ancestral population. So the gene pool of the island population, because it's a subsample and because it's very small, is not necessarily going to be as similar or similar at all to what was on the mainland. You're taking a sampling out of the population, you're putting them on an island, you're allowing them to breed, and so the gene pool changes very, very quickly. Random chance events which lead to those individuals being out on the island. And so population bottleneck is another example, or just an explanation for how the cheetah example works. You take the population that consists of 100,000 individuals, you knock them back to 5,000 individuals, and chances are the gene pool has changed. It is technically evolution because gene frequencies have changed, but it's not evolution because of an adaptive trait or natural selection. It's a random chance event which occurs and affects how evolution works. Drosophila fruit flies on the Hawaiian Islands have a lot of really interesting and unique behaviors that you don't see in Drosophila in other parts of the world. There are predatory Drosophila that actually eat other insects. Very, very rare in this particular group, but they occur in Hawaii. They are a descendant group from an ancestral populating event in Hawaii. Founder events happening there. You can also have adaptation or changing to fit the environment, leading to other types of isolating mechanisms like in these anoles. Anoles are relatively small, pretty quick and pretty agile lizards that tend to live in trees and bushes and shrubs and those kinds of things. And the males have this little sort of a flap down here that's called a dewlap. Right? So a dewlap is what they're going to use to help try to scare off predators. They're going to use it to attract females and try to convince those females to mate with them because they have a very large and colorful dewlap. It's a way of males advertising their presence and their quality to females. So because they live in different environments, they've adapted differently to those environments and they start to look different. The dewlap color changes in response to the selective pressure in the environment. That will then have a secondary effect of influencing female mate choice. So males in one habitat that have a yellowish colored dewlap that's this shape may not attract females from the species down here, which has a dark red dewlap and spots on it. Or this one over here, which is a much larger dewlap with more of a dull brownish color. They're only going to attract members of the same sex and the same species if they have the right coloration and the right phenotype. That phenotype didn't change in response to selective pressure by mates. It changed in response to other aspects of their environment. The food they were trying to catch, the predators they were trying to escape from, the habitat they lived in. That's what influences dewlap color. And dewlap color then influences mate choice. So you get divergence within even a very small habitat based on female mate choice. All right. So when it comes to allopatric speciation, we have a situation where two populations of the same species are going to diverge. That's step number one. And secondly, when they're diverged, when they're separated, when they're isolated, they're going to develop reproductive isolation. So that's the second part of the process. You have to isolate populations because if they're together, how are boundaries going to evolve? How are reproductive isolating mechanisms going to happen if they occur together and if gene flow is complete between the two populations? So we've got to isolate them from each other physically or geographically. And then there also has to be enough difference in the two different habitats so that both populations will develop differences between the other populations. It could be something as big as an ocean. It could be something as small as a river. It could be a field. It could be not really even an island per se as far as an island, being a body of land out in the water. It could be a habitat island on land. Forest species here will not go out here in the open very much. The other forest stand over here would be more or less isolated from the one over here because species don't like being out in the open necessarily. Gene flow, which again is where you have genes or alleles from one population going to another population, tend to slow down or reverse the effects of geographic isolation. So when species are still able to breed together back and forth, any differences that accumulate in the subpopulations will tend to be lost. They'll be washed out when these species breed across those physical boundaries. So speciation in geographically isolated populations is the norm. It's most likely the most common way isolation happens, although there will be a couple of other ways that we'll see here in a little bit.